everyone, welcome back to our Biotechnology channels. For today's video, we'll learn deeper about L-asparaginase productions. Before we start, please meet our crew who will guide you through today's lessons. Now, are you ready to know more about how L-asparaginase can be produced? Let's get started.
First of all, for your background information, what exactly is L-asparaginase? It is an enzyme that is able to catalyze the hydrolysis of amino acid L-asparagine to aspartic acid and ammonia. The addition of water to the L-asparagine structure causes ammonia to fall out, thus creating a structure of aspartic acid. The enzyme is most famous for its success as a treatment for acute lymphoblastic leukemia, a type of cancer. How does it work? Cancer cells need asparagine to grow, but unlike normal cells, cancer cells cannot synthesize and produce asparagine to fulfill their needs. The presence of L-asparaginase helps to convert the circulating asparagine, thereby depriving cancer cells of a key nutrient and causing them to die. Next, what are the steps to produce L-asparaginase?
The first step is upstream processing, which is defined as a production step of bioprocessing where cells are grown to the desired quantity of bioreactors. It is usually done in the lab. In this process, several steps need to be done, such as cell isolation, cell cultivation, and media preparation. In order to get a maximum result, the cultivation process needs to be done under optimal conditions for the microbes' growth, such as pH, temperature, fermentation condition, and nutrient needs. Submerged fermentation is a type of cultivation process that occurs in the liquid broth medium. The broth provides the nutrient for the microbes to develop. Submerged fermentation is the most often used technique to produce the enzyme. The method usually occurs in the petri dish medium. On the other hand, solid-state fermentation is a cultivation method that uses a solid substrate for microbial nutrition source. In this type of fermentation, the moisture content and water source are limited, so the bacteria can only rely on the nutrient from the solid substrate for development. There are several substrates that are used, such as wheat bran, coconut oil cake, black sesame oil cake, soybean, and sugarcane bagasse.
After the upstream process, the production continues with downstream processing. What is downstream processing? It is defined as the recovery and purification of biochemical products. It is a necessary step in order to obtain the product purified form. Therefore, to maximize the result, the downstream process requires several key steps, such as separation, purification, and formulation. There are many techniques and methods that can be used for each step, but for today, let's just study a few ones. One of the purification technique is precipitation. The process started by adding a precipitate agent to crude extract, which is a solution that has been obtained after cell debris removal. For L-asparaginase, the common precipitate agents are ammonium sulfate or methanol. The addition of precipitate agent causes disruption, thus resulting in protein aggregation, which make the fractions fall out from the solution. The step is then followed by centrifugation to collect fractions and form a pellet. The pellet that now contains the target product is washed and dissolved in a buffer. Another purification method is chromatography. It is a technique that is used to separate the components contained in a mixture. It works by having the mixture go through two phases, which are mobile phase and stationary phase. The mixture is first moved by the mobile phase through the stationary phase. When this process occurs, the components in the mixture are separated and create bands, as the components in the mixture have different attraction to the phases. This method is famous as it can produce a high purification value.
Moving on the next step, which is formulations. Another key step in improving a long and short-term storage of the enzyme L-asparaginase formulations is freeze drying. These various names of these procedures including lyophilizations and cryodesiccation approaches. So why do we choose freeze drying formulations? It's because we want to keep the enzyme activity and stability during storage and distributions. And how does these procedures work? It's removing the water content in the frozen materials or a product without going through the liquid phase first.
Nowadays, industrial production is carried out using Aspergillosis and Erwinia chrysanthemi. So researchers have conducted new studies aiming to enhance the production and search other microorganisms that can act as L-asparaginase source, like other bacteria, strains, fungi, and yeast. Some examples of bacteria that have quite high L-asparaginase enzyme activity during their upstream processing is Erwinia arida with 1250 unit per grams enzyme activity rate using submerged fermentation, and P. aeruginosa with an enzyme activity rate of 1900 international unit per milligram using solid substance fermentation. Who knows what the future can bring? Maybe we can get another source that can effectively produce L-asparaginase and is safe enough to be used as medical field. And we have reached the end of our short brief about L-asparaginase production. Do comment what your thoughts are on the subject and feel free to comment down below. A like the video would be appreciated and wanna subscribe while you're at it. Thank you.